Hi, Manish. Good evening.
Good evening.
Thank you very much for accepting our invitation to participate as a speaker here. So it's a great honor to have you here.
My pleasure.
I
How are
will
things
switch
in
to
Brazil?
Portuguese now.
How's the COVID treating you?
Oh, well, thanks God so far. You guys are already getting vaccinated, right?
Well, yes, not
Close.
me, but there
Right.
will be people. Yes, the ICU and the emergency room first, starting
Right.
tomorrow, and then in the next few weeks, more of the hospital. Eventually, more outpatient doctors like me, probably in January.
Okay. In January still? Good, good, good. We have to wait a little bit more longer here, but we'll get there.
Yeah.
So I've been hanging so far. Hope I'll get there. Thank you. So I'll just switch to Portuguese for a few seconds just to introduce you. Olá, pessoal, boa noite. Temos aqui o prazer de ter o Dr. Manish Butte. O Dr. Manish tem um currículo excepcional. Tá? Atualmente, ele é o professor associado com tenure do Departamento de Pediatria da UCLA e o chefe da divisão de Imunologia, Alergia e Reumatologia. Tá? A trajetória dele é muito peculiar, porque ele fez física inicialmente, depois ele entrou na Brown para fazer o seu curso de medicina, tá? Estudou biologia estrutural na UCSF e se graduou com MD, PhD em biofísica, também o PhD, tá? Depois ele foi e completou a residência no Children's Hospital Philadelphia, CHOP, em pediatria, e fez um fellowship, uma residência, né, aqui do Brasil equivalente, em alergia e imunologia no Boston Children's. Depois ele foi em frente e fez seu pós-doc em Harvard, trabalhando em linfócitos-T, ficou nos anos iniciais de faculty at Stanford, e agora em 2016, onde ele assumiu esses cargos, se mudou para a UCLA, onde continua até agora. Ele tem um laboratório, né, e uma clínica, um pesquisador que vê pacientes no seu dia a dia, que é um centro GF Model, é um co-investigador da Undiagnosed Diseases Network, que é financiado pelo NIH, foi começado lá no NIH alguns anos atrás, e um dos cofundadores, diretores cofundadores do Centro Californiano para Doenças Raras, tá? Então, temos aqui um grande prazer de ter o Dr. Manish falando de transcriptoma, ou transcriptômica, em doenças comuns e doenças do sistema immunológico. Manish, thank you very much again, and the stage is all yours.
Thank you. Okay, I'm going to try to share the screen here, just to make sure that
I'll disappear for a minute.
Hang on. Oh, it's the application.
Yeah, once you share, I can add to this stream here.
How does that look?
Oh, it's looking perfect.
Does it look full screen?
Remove myself from the scene. Thank you.
Okay. Thank you, Bosco. Thank you all for your kind staying up late to hear a little talk about immunology and about genetic rare immune diseases. I'm very honored to give you a little glimpse about how we tackle rare immune diseases here, and hopefully we'll be able to work together in the future to take care of some very difficult cases as they come up throughout Brazil. I understand that the technology, especially through groups like yours, to do whole exome sequencing is now flourishing in Brazil, and this is great news. I think what we'll find, and what I'll share with you today, is that even with exome sequencing, sometimes there are some difficulties in making a diagnosis. And so what I'll really try to get today is to share with you is how we now tackle some of those more difficult cases. So if you are ever in Los Angeles, please come visit. Can you see my pointer? If it's on the beach here? Yes, anyone? This is UCLA. We're located. Yes, it's okay?
Yes.
Okay. This is UCLA. We're in the western half of Los Angeles, right against the beach. This is the Pacific Ocean here. This is Santa Monica. This is where I live. And this is UCLA. And you can see it's right next to these mountains. We have beautiful parks and trails for hiking up and down the mountains. And then here's Beverly Hills, where all the fancy people live. So all of you, please let me know if you're ever in LA. We'd love to host you at UCLA and hear about the kind of work you're doing. I don't really have any major conflicts of interest. I'm going to tell you about the off-label use of 2 drugs. And now we are working with the companies that make those drugs to actually try to run a clinical trial. But let me jump right in. As an immunologist, we're often asked, what does it mean for a person to have a defective immune system? When is it worth investigating to understand if somebody has an infection? Is there a defect of the immune system? And to try to really get at the idea of defects, we flip it around to say, what does it mean to have a healthy immune system? What is it to have a competent immune system? So this is an arbitrary definition, but it helps us ground ourselves into the reality of what is healthy and what is perhaps not. So a competent immune system is one that, upon an infection, the body heals itself without major symptoms, but sometimes with symptoms. And in the end, the recovery back to baseline is back to self and without any specific treatments needed. For example, ear infections that happen in the middle ear, typically viral or bacterial infections, can be cured without any antibiotics, without any treatments at all. 97% of the time, we know that antibiotics aren't needed. That's an example of immune competence to those kinds of organisms, to those kinds of infections. Okay, the corollary of that then is that anyone who dies of an infection or has severe consequences of infection where they don't return back to baseline, that is an immune deficiency. And those are the cases that we are most interested in here. Now, you may be surprised to learn, and I was certainly, that most of the serious infections then that cause people to die do not actually kill that many people. For example, take malaria, a major killer of humans. 10 million cases of malaria per year in countries like India, but only a tiny fraction of individuals actually die from a wave of malaria that may go through a village or a town. Similarly, tuberculosis, a major killer of humans, causes TB only in about 10% of people who are exposed to it, and the ones who die from it are only about 2% of those, which means 0.2% of the patients who really are getting exposed to TB are actually dying from it. Similarly, Spanish flu, the greatest killer of humans, of any one infection at any time in humans, 3% of people at most have died from it, although secondary infections are actually attributed for a major portion of these. Staph, strep, candida, and even COVID-19, all much lower rates of death at the level of 0.1% at most of the people who get infected are dying from these infections. Okay, so why are some people dying of these infections then, and other people are susceptible? What is the cause of the variability in between individuals for infection? Well, the most common causes are attributed to things that you can point to easily, like malnourishment. Maybe these people in this village are not fed well, and that's why they have a greater susceptibility to some infections. But we know that even if you take a population of malnourished people, only a tiny portion of them actually die. So it's true that malnourishment leads to an immune defect, but it's not likely to be a significant cause of inter-individual variability. What about the microbial variation itself during infections, during outbreaks? This is probably not relevant. But year to year to year and decade to decade, yes, pathogens can vary, some being more severe than others. But during any one outbreak, for example, during COVID, we don't expect the reason that some people are dying from it in 14 days and other people, 60, 70, 80% of people don't show any symptoms at all. That is probably not due to variations of the pathogen itself between those individuals. And the level of inoculum, certainly a very good hypothesis to explain why some people have more severe disease than others. But we know in examples where patients are infected accidentally with direct injections of perfectly controlled doses of tuberculosis, like in a situation where TB was substituted for BCG in a vaccine, that even in these situations where 250 infants were infected, only a quarter of them actually went on to die. 3/4 of them injected with the same amount of live tuberculosis did not have any severe outcomes. So the conclusion that we draw is that the immune system is quite successful at protecting our species at a populational level. But on any individual level, maybe it's not such a great instrument of protection. Certainly, the majority of people will live any significant outbreak, but some minority number of people will not survive. The immune system is not tuned to make each of us have survived across any infection, but rather is tuned for our species to survive. Now, the impact of our immune deficiency is not really seen on a day-to-day basis anymore, because we have masked the death rate in humans with progress, with things like clean air and clean water. And certainly that has transformed us from the prehistoric era, where the average lifespan was about 24 years, to and in fact, almost half the people were dying by age 5 from infection, to the current world where our lifespans are in excess of 70 years and 80 years on average. So even though we may be predisposed to infections on an individual level, we don't get to see it because we've largely protected ourselves from the exposure to these infections. Nonetheless, if you want to dive down to understand what is the cause of variability in infections and outcomes between people, we really can only have one answer, which is the host genetics. And there are a variety of approaches that have come to that we've seen this result in, including twin studies where monozygotic twins versus dizygotic twins, ones who share the same genome versus not, have a very high concordance rate of death from tuberculosis as compared with the dizygotic ones. And in adoption studies where adopted individuals who live their entire lives outside of the home have a much higher chance of dying from infection if their parents died of infection versus them dying of any other cause, including cancer, if their parents died of that cause, including cancer. It's only infection that seems to have such a high tie to the genetic, with a genetic basis. Now, when we think about the genetic basis of susceptibility to infection, we can really spread our thinking into a spectrum of conditions. Everything from unconventional phenotypes, where there are common variations in genes, things that 1, 2, 3, 4% of all people have, that themselves probably do not impart as a result a major defect, that themselves, because the pathogens that make that susceptible to are relatively rare, you're unlikely to have a major risk in terms of lifespan. Or contrast that with what we call conventional immune deficiencies, ones where there are rare genes, rare variants in genes, like BTK deficiency that leads to loss of antibody function. In these situations, there's typically, because of the rareness and the impact on the gene, a high functional consequence to those variants, leading to loss of function like loss of antibodies or loss of cellular immunity. The phenotype is quite penetrant. These patients show up in emergency rooms and in clinics with infections because they have such a major functional defect. But on a populational level, this is unlikely to cause a significant portion of death or morbidity because the variants themselves are so rare. They occur rarely among populations. So these actually are not two bins. There's a continuous spectrum of these kinds of defects, everything from common variants that may affect many people, all the way to the ultra rare. The take home, though, from all of this is that we have done a very good job in the world of immune deficiency, genetic immune deficiencies, to convince people that those at the very high end of the bell curve, the ones with the very highest, most severe phenotypes, probably don't have bad luck, but instead have a genetic basis to their disease. But the really important point now is not just to think about those most severe cases, but think about the ones where all infections may represent a defect of immunity. Not just the penetrant ones, not just the ones that are Mendelian, which means monogenic inheritance with high penetrance, not just the most severe cases, not just the ones with weird opportunistic infections. Any infection where the host doesn't return back to baseline probably has a genetic basis. And part of our worldview has to shift in the next few years from working very hard to discover these rare genes and these rare alleles leading to ultra-rare immune deficiencies, and instead start thinking about the common variants in here that could be contributing to disease phenotypes. Why is this relevant? Because genome sequencing is cheap. It's become very cheap now, from, of course, billions of dollars 20 years ago to now under $1,000. We're typically doing genomes in our institution with results coming back in about 2 weeks and costing about $700 US. What led to this big decrease in costs from the greater than 1,000 to less than 1,000 era? Largely, it's the inventions from Illumina of short-read sequencing at high scale, and especially in the last 3 years, this instrument called the Illumina NovaSeq 6000, which provides enormous scale and allows $700 genomes. Now, of course, this is a very costly instrument, not something you're likely to have in your home, but something that can really transform genetics when an institution has one. The relevance here, of course, is beyond just instruments and costs of tests, but really, how do we impact patient care? We now, because of the low cost, believe that genetic testing should be the standard of care for all rare diseases. Certain countries like England have passed, and Canada have passed guidance now with their nationalized health systems to mandate genetic testing for all patients with rare diseases. In the United States, we don't have that opportunity. We still have to deal with private insurance companies as being the major barrier for getting genetic testing. And why are we doing genetic testing? It's because it really helps patients fundamentally. They can end their diagnostic odyssey. Patients who are spending years or decades trying to figure out why they are sick can have an answer. It also can avoid some unnecessary testing. Patients who were going from doctor to doctor trying to get test from test. Instead, that can be aborted and the answer can be given to them. In our world, where private payers are not interested in funding treatments or testing, having a genetic diagnosis is an ace card to play, where in that card game against your payer, you can say, I have this BTK mutation, my B cells are low. They're not going to be able to argue with that as the basis of your disease. And what we're really trying to get then is moving patients into specific targeted treatments. Everything from immune globulin replacement to gene therapies, which are very expensive in our society and require insurance approvals to move forward. We've also started to use repurposing drugs from the cancer world and from the autoimmunity world to start treating our genetic diseases. And these drugs are very expensive. We need to have the right genetic basis for disease in order to target the pathways that have become disrupted. Finally, of course, genetic testing allows for family planning, genetic counseling, things that groups like yours know very well are actually things that many patients are desperate for. I just saw a 36-year-old in clinic a few minutes ago who's desperate to have another child and really wants to be sure that her child doesn't have the same immune deficiency that she does. Okay, when we talk about genetic testing, of course, I really don't need to tell you that we can be sequencing on the genomic DNA, of course, the exons, the coding portions of the genome, and the introns, the intergenic parts or intronic parts between exons that do not code for protein sequences. These are converted through the central dogma to RNA, and these exons are joined together to form a message, and the message codes for all the proteins of the immune system, everything from antibodies to soluble cytokines to the cell surface receptors that are important for signaling. When we do exome sequencing, of course, we're sequencing the exonic portions of the genome. And that has worked very well for us for about 6 years on a conventional basis to sequence just the exons, either through targeted panels or through exome, which is a larger targeted panel. But we've gone further than that. And really the point here is to tell you about some of these further innovations. We've moved from just looking at the coding portions of the genome, which make up about 1%, 1 to 2% of the genome, and instead start focusing on these intronic portions of the genome, which make up about 1/3 of the genome. The other parts are these intergenic regions between genes, which we still don't have a very good way of analyzing or understanding. So the advance that I'm going to tell you about today really helps us understand not just the coding portions, but also these intronic portions. Okay, so remember that the intronic portions come together, the exonic portions come together to form a transcript, and the transcript leads to the protein, which may or may not have a functional consequence. The philosophy I'm going to tell you about really involves this sequencing down here at the RNA level, at the mRNA level, rather than up here at the genome level. Sequencing at the RNA level allows us to identify not just the coding portions of the genome, but also if these exons were put together properly. Okay, so what does that mean? We actually combine genome sequencing and RNA sequencing here, and that is the way that we really try to approach both exonic variants with high coverage and with really good, so almost tiling level coverage across all the exons, but also look at the impact of the intronic variation by looking at the impact on splicing. For example, if there's a genomic variant that lies solidly in an intron, 100 bases away from the nearest exon, that impact of that variant in the intron may actually affect the splicing pattern, which you would see on transcriptome sequencing. That means if you can put together the mRNA properly with short-read sequencing by looking at these junctions, for example, which I'll show you in a second, that allows you to really identify whether this exon is being spliced properly or improperly, and thus understand, is this intronic variant the cause of a rare disease? Another thing that sequencing genomes does is allows us to identify whether there are variants in coding regions that can tell us about allele-specific expression. That's important because if there is a variant in a coding in part of the coding sequence that doesn't obviously change the function of the protein from first glance, but instead changes how much expression that allele has, this C variant is underrepresented in the transcriptome, whereas the G variant is overrepresented in the transcriptome, then the impact of that variant could be magnified because it makes up such a higher proportion of the reads. Finally, transcriptome sequencing can tell us about the expression levels of genes, which is critical, of course, for proteins to function. You have to be able to make transcripts, and the transcripts have to be able to turn into proteins. If the level of expression of those transcripts is low, then you're not going to make a lot of protein, and that could be also the cause of disease. As we move into RNA sequencing, I'm going to walk you through the steps that we do. And the first and most important is after extracting RNA from cells and capturing all the RNA is to do some quality control, or QC. And typically we are looking at quantities of RNA that must be sufficient to do the sequencing, and then the quality of RNA. And typically we've been using an instrument from Agilent called the bioanalyzer that gives us, based on electrophoresis, the quality of the RNA based on the heights and the widths of these peaks. If there's a lot of degradation of RNA into smaller forms, then it'll show up on plots like this. And the single numerical number, 3, 5, 10 here, tells you the overall quality of the RNA. It's very important to have high quality RNA if you're going to do transcriptome sequencing. The next thing you do when you're doing RNA sequencing is prepare a library. The libraries are needed to go onto the Illumina instrument, but you have to know what you're preparing a library from. In cells, there are all kinds of RNAs, non-coding RNAs, small RNAs like microRNAs, ribosomal RNAs that code for the functional parts of the ribosome, and of course the messages themselves. Most of the time we're interested in messages. And so we have a way of capturing these RNAs from the rest of the pool, things like poly-A selection, or called poly-A prep, that really captures just the messages and not the rest of the parts. Or ribodepletion, which gets rid of all the ribosomal RNAs, which make up actually over half of the RNAs in some cells. And then, of course, size selection, to get rid of the small RNAs if you're interested, or capture the small RNAs if that's something you're interested in. Once you've gotten the right portions of RNA, then you can convert them to cDNA through reverse transcriptase and put primers on the ends and drop them into an Illumina sequencing reaction. Of course, it's important to then understand if the transcript you're looking for is expressed in the tissue that you're going to actually be analyzing. Drawing blood from individuals is certainly useful for doing genome sequencing, but is drawing blood the best place to find transcriptome? Maybe not. And so the place we look for this is in databases like GTEx, which is a set of 54 tissues from 1,000 healthy individuals that just gives a general idea of how much each transcript in the body is expressed in those tissues. For example, this transcript called CAT6A is expressed in high level in the lungs. You can see the bar here, this green little bar here is up high. However, it's expressed very low in the brain and very low in heart tissues. If you were to be taking an endomyocardial biopsy from a patient and then say, does this patient have CAT6A, you're unlikely, variants in CAT6A splicing, you're unlikely to see an impact if you're looking at heart tissue. Maybe you'll see an impact in blood. Wow, you'll see a good impact if you're looking at lung biopsies. Does that make sense? So you have to really capture not just if the transcript is being expressed, but which transcript is being expressed in which tissues. And this database, GTEx, provides a really great resource for that. Okay, so once you've decided to get the tissue that you really need, in immunology, we typically draw blood because all the immune cells are in blood. And then we sort sometimes the cells we're interested in, like T cells or B cells, away from all the other white blood cells, and then do our transcriptome from that. B cells are captured here in GTEx here, in this little purple bar here, where it's EBV transformed B cells or lymphocytes. Okay, so once you've captured the tissue you're interested in and you have done the quality control and the selection of RNAs, then you proceed to sequencing. And then you try to, we're going to talk about how we assemble the data and do analyses. Okay, so what are we looking for? Again, we're looking for alterations in splicing. If these are our exons, blue, orange, yellow, and green, and transcription flows in this direction, then we would like to see the transcription in this pattern, blue to orange, orange to yellow, yellow to green. And the junctions between these exons, if we do reads across this area, should reflect this sequence of exons. On the other hand, sometimes we find something called exon skipping, where there may be a variant in a particular site that leads to skipping over the orange exon. That, for example, will change the message. And the junctions from blue, instead of going to orange in the transcriptome, will actually go from blue to yellow. That's the hint that there has been exon skipping. We also see exon retraction, where you may see a portion of the exon, being captured, but a portion of the exon being lost. Here, for example, the reads across the blue-orange junctions do not go from the edge of blue to the edge of orange, but instead go into the middle of orange. That tells you that a splice acceptor site perhaps has been created within this region, sometimes a synonymous mutation, and that causes a change in the pattern here. This can lead to frameshift mutations and other sort of highly deleterious damage to the transcript. I just saw a patient with this a few days ago with an immune deficiency where there was a synonymous mutation in the gene that led to catastrophic loss of expression of the protein. This is in a patient with a cellular immune defect. Similarly, we could see exon extension, where there may be a change in these splice acceptors. Instead of the canonical one here, there may be a new one, and that new one leads to a growth of the exon into the intronic region. Finally, you could see pseudo-creation of exons, where there may be a new exon created because of an acceptor splice donor gain here, or splice acceptor gain, and you end up with a new portion of intronic data inside of the orange-yellow junctions. This obviously is not good for the message if this leads to garbage being inserted into the protein. Intron retention is a bigger consequence of this, where the entire intron may be retained in the message, and that can be very deleterious as well, introducing both weird protein sequence, or also frame shifts. Okay, so these are all the kinds of things I'm talking about, where instead of getting this normal message, you have exon skipping, or retraction, or extension, or pseudo exon creation, or intron retention. And again, how many bases are added or eliminated is really why transcriptome sequencing turns out to be so important for identifying disease. Okay, so how do we analyze things? We take on an RNA-seq first approach, because genome sequencing contains tens of thousands, if not hundreds of thousands, of rare variants. And many of them are so benign that it's impossible to go through tens of thousands of genes, hundreds of thousands of variants to understand which ones are significant or not. And on the other hand, if you can dive into RNA sequencing first and look for which ones have an observable effect on transcription by looking at defective transcription, then you can narrow your list of genes down to just those ones that have aberrant transcription going on. So we do filtering, of course, by frequency across a cohort after identifying all aberrant splicing. And then we'll look in the genome to see if there are rare variants genomically that explain the rare splicing consequence. Okay, so the pipeline is very straightforward. We get RNA typically from tissues of interest. Immunologists get it from blood. And then we do RNA sequencing, as I mentioned, after doing our selection for messages. We run it through very standard tools like STAR and splicing tools from things like Cigar, and very off-the-shelf tools, including some custom scripts that we have to run all this work in a straightforward fashion. We calculate frequency of these exon-exon junctions and look for protein-altering potential. And then in the end, we make a list. And these table of rare splice variants then gives us information about whether a rare splicing is going to be phenotypically, biologically relevant. Okay, things like splicing ratio, which I'll show in a second, the frequency in other family members turns out to be really useful. And then looking at things like intolerance metrics, how tolerant across a large ensemble population are rare variants. Of course, in the end, we really need to be able to attribute this gene to a phenotype. And if it's a gene in a pathway that's already understood that really supports the phenotype we're seeing, for example, a gene in a pathway for B cells in a patient who lacks B cells, then that's going to make a much better story as we look at these tables of variants that come up. Okay, so how do we, here's an example of annotation in a cartoon. You know, we look at junctions. The read here started here and transitioned here. This read started here and ended here. This read started here and ended here, etc. And we always try to take canonical annotations if we can. The start of the junction and the stop of the junction. If both of them are annotated, and then this is a canonical junction. If both ends are part of different annotated splice junctions, that could indicate an altered splicing. For example, to go from this end of exon to the start of this exon, as we see with this read, that could indicate dropout of this exon, or it could be a canonical splice that's seen in a different tissue. So we always have to be cognizant about which tissues we're looking at and what the expected splicing patterns will be. Sometimes, of course, we see where neither junction is annotated or expected. This might be the creation of a neoexon in tissues that we would not have in DNA that we would not have expected. Okay, so when we see then an end of an exon lead to a new junction, then rather than looking here where the junctions are all annotated, here we see that this junction is not annotated. This exon, in fact, falls in intronic space. At that point, we know that this is an example of exon extension. Exon skipping, similarly, where you can see that the canonical exon has been dropped out. Exon creation tells you can see that when you see reads occurring multiply inside of a portion of intronic space that you didn't expect. And how do we tell some of these things? One way is by looking at a ratio of the novel junctions to the total junctions. For example, this junction to this junction is expected, and we had 5 reads for these. But we also had 5 reads that went from a novel junction. So here, 5 new reads divided by 10 total reads leading to this exon is a ratio of 0.5. We're really interested if anything is greater than 0.1. Those are often novel enough that we need to dive in further and think about what's going on. This is a quick way of filtering, though. If you see a few rare reads in a novel area, not enough to even make 10% of the total reads at that junction, then you can usually skip them. Okay, here's an example of a case of a 13-year-old with developmental delay who was seen in our undiagnosed diseases network and who had a negative genome sequence. We found a splice variant, on the other hand, in a gene that was very intolerant of variation. This is a high PLI score. And transcriptome sequencing showed that this patient, the proband, had creation of a neoexon here, 1,000 bases into the intron, whereas the parents did not show any junctions in this region. And the splicing ratio here in terms of neo-exon splicing versus traditional canonical was 0.14. This was enough to explain then that these aberrant transcripts were being formed, and that led to loss of function I think in this case, a loss of function frameshift variant that explained the phenotype based on eyes and dysmorphology and developmental delay, and we were able to make a diagnosis. Okay, intron retention is sometimes hard to pick up because some pre-spliced mRNAs will be showing up in your exome, in your RNA sequencing. And how do you decide whether it's pre-spliced mRNA versus a defective intron retention? The way we do that is by counting up the number of reads at the exon bounds and then also the ones on the inside in the intron. And we hope that the ratio of intron to total reads is not particularly high, in which case that might be a sign of intron retention. Usually we want these reads that span junctions, span introns from exon to exon to be much greater in population than the ones that lie in the intron. Here's an example of reads that from a synonymous mutation. Here you can see lots and lots of reads that have fell in this otherwise intronic region in a gene called SARS2, and that led to here's the intron retention event seen in the patient's fibroblasts, the siblings' fibroblasts, and the father's fibroblasts. You see an intron here, whereas in the mother, you see no reads in this intron. This represents a variant that was inherited from the father into 2 children, leading to retention of intron 15 and deleterious loss of function. Okay, so thinking about this kind of RNA sequencing, it really does lead us beyond the era of exome sequencing, where we expect to solve less than 1/3 of cases. In the Texas Children's data set, in our data set, and many others, we found that only about 25 to 30% of exomes are diagnostic, even with trios. Whereas with whole genome sequencing, we could typically add 5 to 10%, sometimes as much as 15%, because you're capturing a lot of other exons that aren't well covered by exome. You're finding new genes potentially. You're looking for structural variation that wasn't possible with exome sequencing. And so we typically get a nice boost. However, when you add in genome sequencing plus transcriptome sequencing, so far we found that a diagnostic rate goes up by another 15%, and maybe as much as 20%. This is taking us all the way up to the 50 to 60% line for diagnosis rates. Much better, almost double what we were getting with clinical exome sequencing, even with trios. This is why we do genome RNA sequencing for our rare Mendelian diseases at UCLA. Okay, I'm gonna take a pause for a second, see if there's any questions before jumping into a case for the second half of the talk. I can't see anybody, so I hope you'll shout out if you're able to. Otherwise, we'll take questions.
No, you can't see anybody, yeah. But this is very nice. I don't know. Do you want to wait for until the end?
Yeah. We'll do questions at the end. That's fine.
Yeah, because I do have a film.
Okay. All right. Okay. So I'm going to talk about a case. This is a case of a 4-year-old boy who came to our clinic in February of 2 years ago. He came in with this rash that rapidly spread on the back of his neck, and also these lumps on his forehead. They were very tender, very rapidly growing. This grew over the course of about 3 weeks. One of them got biopsied, and the biopsy showed this. This is a spherule. It's a very characteristic lesion that is particularly seen in a fungal infection called Coccidioidomycosis. Now, this is a particularly interesting infection throughout the Americas, and in fact, the very first case was described in this Uruguayan soldier named Domingo Escura, diagnosed in 1889 because he crossed from Uruguay to Argentina looking for a doctor to help him understand why he had this rash on his cheek. It was a severe and painful rash, rapidly spreading. He came to the attention of this doctor, Dr. Alejandro Posadas, who was a surgeon and pathologist, and made some biopsies and found those spherules and thought that he had found a fungal cause to a skin cancer that we call mycosis fungioides. Of course, this was not a skin cancer. This is a severe skin infection, a form of disseminated skin infection that had spread into his skin. This fungus, Coccidioidomycosis, is actually not just in Argentina, but in Uruguay even, but also spreads all the way up through the Americas and into parts of Southern California, Arizona, New Mexico, and parts of Texas up here in America. Other endemic fungal infections, including paracoccidioidomycosis and histoplasmosis, are similar in that they are endemic to different regions based on the geography, the spores that live in the soil, etc. So I had a chance to actually go to Argentina 2 years ago and see Domingo Escura's feet and hands that have also been preserved at the University of Buenos Aires. You can see that his rash spread into the portions of his foot and onto his hands. And all of these are collected in a display in the library there at University of Buenos Aires, including the very first micrographs of this infection and his manuscript that he published in the late 1800s. Coccidioidomycosis is common now in California. There are over 9,000 cases last year, and we spend a lot of money taking care of these cases. On the order of $1 billion in California is spent every year taking care of patients with coccidioidomycosis. In California, it's also called valley fever because it occurs in the Central Valley of California. Some of these cases go on from valley fever to causing disseminated cocci, and I'll talk about that quite a bit. That's what our boy has. There are about 200 cases a year in California, and we spend about $200 million, about $1 million per case to take care of these patients. They are so severely ill. Cocci, in general, is largely asymptomatic, like COVID, like many other infections we know of. Like I talked about in the beginning of the talk, the majority of patients with most of these infections have very few symptoms or no symptoms at all, and Cocci is no exception. About a third of the patients get valley fever, a chronic cough that leads to a self-limited fungal pneumonia. Some go on to having more severe forms of lung disease, but about 1% go on to having dissemination outside of the lungs to bone, to skin, to the meninges. And many of these cases are rapid and fatal. There are about, as I mentioned, 200 cases in California a year. These are often described to occur in states of immune suppression, like in third trimester of pregnancy, when type 2 immunity is dominant, during immunosuppression from transplants and cancer, after HIV infection. And then there's also these mythologies, like they occur more in African Americans and Filipinos, which we do not have any genetic basis to believe that these are disease or states of being at all. But the mythology persists, and we're trying to understand what it means. Of course, there are patients with genetic immune diseases that have been described, and these provide a hint as to what the underlying immune defect is. In the 8 patients who've been described with immune deficiencies leading to Cocci, collected by Steve Holland's group just a couple years ago, we know that 6 of them had defects in what's called type 1 immunity, an inability to produce or signal through interferon gamma, either due to defects in IL-12 receptor that's needed to make interferon gamma producing cells, or in signaling of interferon gamma through STAT1 or the interferon gamma receptor. 2 of these patients had not decreases of type 2 immunity, but not decreases of type 1 immunity, as you see here, but excessive type 2 immunity. These are patients with monogenic defects in STAT3, dominant loss of function defects that lead to what's called the hyper-IgE syndrome. Patients who have excess eosinophilia and IgE levels that we know are potentially an inspiration to understand what is the common cause, what are the causes of Cocci dissemination. In fact, excessive type 2 immunity has been known for decades now. Patients who have excessive IgEs or eosinophilia, or even just generally IgEs, have a higher level of having disseminated extrapulmonary disease. So why is type 2 immunity a risk? Typically because type 2 cytokines like IL-4 and IL-13 tell macrophages to switch to a transcriptional program called type 2 macrophages or M2 macrophages. And instead of having the ability to fight microbes and kill, instead they actually have a more homeostatic function and anti-inflammatory function and are unable to kill fungi. So our hypothesis is that dysregulated immunity, either through defects of type 1 or excess of type 2 immunity, lead to dissemination. Okay, let's go back to our case. This boy had a PET scan, and you saw lesions all over his scalp, his wrist, his tibia, his sternum. This was one of the lesions on his wrist. This area was very tender, but we did not see any particular lesions here. However, in just a few days, this came out of his skin, an extremely tender and painful, destructive lesion, destroying and eating bone and tissue and everything in its path. This is an extraordinarily destructive fungus. It eats everything. And he was able to get these removed through a series of surgeries, this poor kid. Lesions on his forehead. We had to have a surgery to remove one of these, but lesions in his ribs, in his clavicle, and even in his vertebral body that led to destruction of the vertebrae and collapse and scoliosis were rampantly happening in this patient despite being on triple antifungals. We were unable to get this disease under control. Now, inspired by Steve Holland's paper, we looked for signaling in interferon gamma. We saw no defects. We looked in signaling of IL-12, and we found a hint. Giving whole blood IL-12 and looking for phosphorylation of STAT4, we found very low signaling of IL-12. That's a very important hint because IL-12 is needed to teach helper T cells to become Th1. And Th1 cells make interferon gamma, and interferon gamma, as I mentioned, is important for telling your immune system to make the proper kinds of macrophages to fight infections. So if he has low IL-12, we would expect to see low Th1 cells. And what do we expect to see typically in patients? Ratios of 4-to-1, 5-to-1, 8-to-1, Th1s to Th2s. So we went looking at his Th1s and Th2s, and what we saw is, to our surprise, only about 4% of his T cells were Th1 and 35% were Th2s. This is a 9-to-1 ratio in the wrong direction, whereas in a healthy control, you'll see, as you expect, more Th1 than Th2. So Th1 deficiency means he's not making interferon gamma. We gave him interferon gamma. This is being given off-label, and it worked very well for slowing down his inflammation. In fact, we saw that his inflammatory marker, like C-reactive protein, plummeted upon starting interferon gamma. Some of his lesions got better. For example, the lesion on the front of his forehead largely went away. This one over here changed a little bit and coalesced, but didn't go away. And this one on the side in his temporal region actually got bigger. So we really found that interferon gamma could slow down the trajectory of his disease, but not halt it. This is what his lesion looked like, the one that was coalesced, and it started to separate through his skin, an extraordinarily painful lesion on his skull. The pain here was so severe that he was unable to eat. He had headaches and was unable to eat. We had to feed him through a nasogastric tube. This was a very dire time for him, being on triple antifungals and interferon gamma and still not getting better. We went back to our T cells and asked them, if you're making so many Th2 cells and Th2 cells are making so much IL-4 and IL-4 blocks Th1 cells, maybe if we can block IL-4, we could increase the amount of Th1 cells he made. We are thinking that because recently in the United States, a drug became available called dupilumab or Dupixent that blocks the IL-4 receptor. And so we thought to ourselves, if that's true, we should be able to rescue some of his Th1 function by blocking IL-4. We tested it in vitro first, and we were able to find that we could take 30% of his T cells to Th1 upon treating them for 1 week with dupilumab. In a healthy control, it was even up to 60%. So this was really a large rescue of his Th1 function. And so we started him on dupilumab. We did not have any way of dosing this medicine in a 4-year-old. We had to make up a dosing regimen by looking at stimulated responses to IL-4. In vitro and see that we were adequately shutting down all IL-4 signaling, and then we waited. And what we saw was that his Th1 to Th2 ratio rose, and his Th2 numbers were falling, and we really brought him to a ratio of 1:1 instead of 9 or 8:1 in the wrong direction. But more importantly than his T cell phenotype, we rescued his function. We eliminated the spinal lesions, and even the lesions on his scalp melted away over the course of a few weeks. This is a picture of that lesion on his forehead just a few days after started dupilumab. This was about 4 weeks later, crusted over and gone. This is another lesion almost totally regressed away. So we believe that these kinds of treatments are general and can be used for all severe fungal, viral, and bacterial infections, not just in the United States, but across the world. There are endemic fungi. In Brazil, for example, as I mentioned, not just Coccidioidomycosis and Coccidioidomycosis, but also other infections, including these regions of high burden and histoplasmosis that are throughout the South America and even through portions of the central parts of the United States and parts of Africa. The philosophy here is that instead of treating infections with just more antifungals and just more antibacterials, we can use genetics and immunology to try to identify pathways that we can change to skew the immune system to also help fight infections. Okay, so what about the genetics? I started this talk talking about genetics. Let's talk about the genetics. Our patient did not have any rare immune deficiency, despite that we found he had almost severe attrition of signaling through IL-12 receptor. However, our philosophy in looking for variants is all about rare diseases, rare variants. And the reality is we shouldn't expect a rare variant for this disease. Why? Because coccidioidomycosis in California, Arizona, New Mexico, Texas represents an endemic infection. But if you go one state away to Oklahoma or Nebraska or Kansas or Louisiana, there is no cocci at all. And so the selective pressure on variants that may be pathogenic, that may create a susceptibility to cocci, there is no selective pressure if you just go a few hundred miles away from where the fungus lives. That means we should not expect a purifying selection to occur on these genes so that the pathogenic variants become rare in a population. We should only expect purifying selection on genes when the selective pressure on genes is universal. If the selective pressure is narrow, then there is no purifying selection. And so our whole philosophy of looking for rare variants in a genetic disease, in a putative genetic disease like this boy has, does not work. And we don't have an answer for this. This is an area that we need to build new tools and new thinking about. How do we go after non-rare variants as we tackle these diseases? We can be inspired, however, by other examples where this has been shown true, including John Casanova's group recently showed that there is a relatively common variant in the signaling molecule called TYK2, a variant that occurs in 1 in 600 Europeans homozygously and is carried by something like 1 to 2% of all Europeans, at least on one allele. This is a pathogenic variant of converting a proline to an alanine in this signaling molecule, and slightly deranges the signaling of IL-23, a sister molecule to IL-12, by the way. This variant leads to susceptibility to tuberculosis. And so even though there is no rare variant here, there's actually a common variant, 1 in 600 we consider pretty common. It can be deleterious to function enough to lead to a significant increase in tuberculosis. Okay, so when we went looking at not at the genome, but at the transcriptome, as I showed you the examples in the beginning of the talk, we made our discovery. What we found is that our patient had 94% of the transcripts went from this exon. The transcription is running from right to left in this gene, from this exon to this exon, and 6% of the reads went from this exon to this exon. Now, this version here is called the long isoform. It says short here. This should be long. Sorry, this is a mistake here. So the long isoform creates a longer signaling molecule. This is taken out of zooming in on a particular region on chromosome 13. I don't know what chromosome this is. Whereas the shorter isoform ends here. And the consequence is that the shorter isoform does not contain the transmembrane domain and the intracellular domain that are key to signaling. Therefore, the short isoform, which our child made 96% of his isoforms were short isoform, is not able to signal. And that explains on the transcriptional basis why IL-12 signaling did not get through, why he did not make Th1s, why he did not make interferon gamma, why he made too much type 2 immunity as a default then, why he skewed his macrophages in the wrong direction, why the fungal infection was able to spread from his lungs all over his body to almost kill him. When we looked at 408 healthy individuals, we found that the transcriptional ratio of short to long took on a distribution like this. And what we found is that the average was actually about 67%, 68% short isoform. That's to say, in you or me, we have mostly short isoforms. That's kind of surprising. I didn't know that, but we basically make short isoforms mostly, and about a third of our isoforms are long. And his sister was that way. Interestingly, his mother also has a significant skewing to short isoforms. We do know the biology of IL-12 receptor changes upon activation, and especially in interferon gamma signaling, to transcribe the longer isoform. And indeed, in our child who survived his infection and stayed on interferon gamma for over a year, we were able to show a year later that he was making now a proper ratio of short to long isoform. No longer 94% short and 4% long, but 60-30, just like we would expect to see. This transcriptional restoration also led to a functional restoration. He could now signal phosphorylation of STAT4 through signaling through IL-12. And when we plot where he appeared, he went from here at the very top of the 1th percentile for aberrant splicing in the wrong direction to here, a patient who had really laid right on the midline. Okay, so this is an example of how transcriptional sequencing is very important for identifying the immunological defect and explaining disease. Now, unfortunately, we don't have a genomic cause for this splicing defect, and we may not find one. This may be epigenetic. This may be something else that we don't see encoded in the genome. We are looking and learning how to look at non-rare variants in this region to see if there's any non-rare variants that could be explaining the splicing. Those are things like splice QTLs, for example, that we know correlate with changes in splicing based on relatively common polymorphisms throughout the coding and non-coding regions of a gene. But we haven't discovered it yet. We still have a lot to learn, but using exome sequencing, genome sequencing, and now genome plus transcriptome sequencing allowed us to come up with an answer for this child, and I think is a very important technique for all of us to move towards in the future years. So I just want to acknowledge Monica Tsai is the clinical fellow who did all the work for the Cocci patient. Tim Thalen's the postdoc, was a postdoc in my lab, is now a project scientist in my lab who did all the immunological testing and All the sequencing was done by expertly by Hein Li, Alden Huang, and Li Kai Wang. These guys also provided a lot of the slides that I showed related to splicing and how we analyze splicing. And these three and myself are all part of the Undiagnosed Diseases Network, which is an NIH-funded group here at UCLA and at 5 other hospitals in the United States that is headed by Stan Nelson and Julian Martinez and Christina Palmer, and then including some genetic counselors and a huge staff of scientists, all of whom are dedicated to figuring out the genomic basis of human diseases. Thank you very much for your attention. I'd love to take some questions.
Thank you, Manish. It was excellent. Thank you very much. I don't know, this case was a great case to, highlight all the power of immunology. Let's say, you, even not knowing the genetic defect, you solve the problem.
Yeah.
I never heard of manipulating only the Th2 pathway with dupilumab, and have such a strong effect on a fungal infection. But of course, gamma have been trying for, in different scenarios for a long time. Steve Holland at the NIH and many others.
Right.
But the dupilumab effect was really interesting.
Yeah, and we're lucky now to have the sponsor of dupilumab, Regeneron, be so excited by this one patient's data that we have been given enough funding to think about doing a clinical trial where we're going to randomize patients to either getting placebo or dupilumab. 2/3, 1/3, I think, will be our ratio, and these will be patients with disseminated cocci in California. We hope that we can repeat what we found here. When we've gone looking at Th1, Th2 ratios in disseminated cocci patients, we found that about 20% of them, not all of them, about 20% of them, we've looked at about 50 of them so far, about 20% of them have that excess skewing of excess type 2 cytokines, X2. So there may be many sub-immune diseases within disseminated cocci, one of which, this portion, this 1/5 of them maybe, has a type 2 disease. And we're hoping those are the ones that will really benefit from dupilumab treatment.
So I would ask, you are going to use this as a selecting factor for treatment with the dupilumab or not? The skewed Th2 to Th1?
We're not going to as an a priori testing, but we are certainly interested in seeing if those are the ones who benefit. Because there's really no targeted therapy for this disease, we don't want to exclude some patients from treatment. Even if they don't have that excess type 2, maybe it's not enough, and we're just thinking anything we can do to push type 1 immunity by blocking type 2 immunity might benefit everyone.
Perfect. Manish, I have a few questions here I see on the board, but I have a couple technical questions with the transcriptome, of course. In our project, we're going to sequence, we just started sequencing 5,000 patients with rare disorders. And one of my, I just set up about a couple NovoSeqs. We have 3 NovoSeqs right now, 2 for my project, for the project.
Wow.
1 for the clinical use, and we just started recruiting from different centers. And that's why I wanted to hear you, because we are planning on doing this transcriptome like a side project inside the Rare Genomes project. And of course, some of the defects or the possible findings are this is exon skipping, exon retention, pseudo-exon inclusion. Some are easier, some are harder to detect. But for most of them, there are a lot of normal splice variants for a gene. So a gene may have its longer form, as you showed, in a shorter form that's not aberrant, right?
Yeah, right.
So for us, looking at immune disorders, you probably run a bunch of controls. So you have your baseline. How did you go about this first, preparing your house to have comparisons later? So you have to create a base first, right? A database.
Yes and no. It turns out that we are able to use whole blood transcriptomes in diseases where we think the phenotype is unrelated as the basis of a controlled data set. For example, in our UDN, many of the patients have been brought in for genome RNA sequencing who have a neuromuscular disease phenotype, patients who have atrophy or skewing of their muscle fibers or something like that, Duchenne's-like diseases, et cetera. In those patients, when we look at whole blood transcriptome and look at IL-12, for example, receptor, we don't think there'll be an IL-12 defect in that patient. So we can learn from their splicing patterns as being potentially part of the controlled data set. So we didn't need to build a database of healthy controls first before starting to look at the aberrancies. That would be too much work, especially given that we know that transcription changes with age, with sex, with all kinds of factors. And by the time you built a healthy data set, you've blown $1 million and nobody has that kind of money. Maybe you do, you bought 3 NovaSeqs. But instead, we just bootstrapped our way forward by looking at the genes in tissues and in individuals where we don't think that they're going to be defective. Admittedly, we could go wrong in that kind of conclusion. But as you get to 30, 40, 50, 60, you have enough data that you can start to ask, okay, well, this guy actually was a little bit of an outlier, even though we didn't expect it, but these other 59 are fine. And you'll find that you have enough data to work with from there. At the same time, there are public databases that you can use, and certainly those are not necessarily well annotated with age, with sex, with all these other things, but they are out there that you can definitely use public databases too, like GTEx.
GTEx, right. And do you always use family members? Do you always run mom and dad, for example, if you have access or a sibling?
Yes. Especially when you move beyond exome and panels into genome, and you start it with so many variants, hundreds of thousands of variants, you go crazy if you can't filter them out by using very simple rules like mom has that variant and mom is unaffected, sibling has that variant and sibling is unaffected. Take it off the table. It's not perfect. We know that lots of diseases have partial penetrance, and you may take things off the table prematurely. But it's a good starting point to start filtering down from the hundreds of thousands down to the few thousand, when you can really start to look at genes and make some sense of them.
Excellent. So, so that's a good approach. I mean, it's actually, you mentioned something about RNA first, right? I was very curious about this. Yeah. So when you said RNA first, is after a whole exome or a panel, or RNA first?
No, it's definitely genome RNA, but the idea is to look at the RNA first and just ask, where are the transcriptional aberrancies?
And then go back to the data.
Then go to the genome to see where they are. Yes, exactly. You got it.
Perfect. Very, very curious.
I see a question from Lucila.
Please go ahead.
Yes. So do you think that coupled genome transcriptome approach can help discover and validate new PID causative genes faster? Yes, exactly right. And in fact, we would love to propose to work with you. If you have unsolved cases, if you're doing genome and transcriptome sequencing, it sounds like you're going to be doing, we'd love to work with you to help understand if you find pathogenic variants in genes that you want to do technical validation around. Anything we can do to help this effort. We do think that the era of exome sequencing will come and go in the near future. Panels seem to be cheap enough and powerful enough and quick enough, a few days, that they're going to serve some utility for the next year or two. And exomes fall, unfortunately, in between. They are expensive enough, $300 or so, where you're getting a lot of data, but not quite enough. Whereas if you just spend $200 or $300 more, maybe $400 more, you're getting the whole genome. And so we see more and more are thinking that we'd rather spend a few $100 more and get the whole genome rather than the exome in between. Exome sequencing in particular has lots of problems in terms of coverage of exons. We're publishing a paper, myself and Sarah Hendrickson from CHOP are publishing a paper soon that looks, and what we found is 27 of the 400 PID genes has significant problems in coverage across all their exons. And so that's a problem when you're going to count on an exome giving you the answer and it doesn't even have good coverage.
Excellent. Oh, I would love to collaborate. We're probably going to have, of course, we expecting a hit rate of 30 to 40%. Yes. Everything else is going to be negative, and those are the cases. I'll contact you later. Maybe we can formally collaborate on this genome transcriptome initiative. But and we are collecting all the data using a phenotypes. So everybody that's recruiting on the different centers in Brazil is filling up all the data already. HPO, in the phenotypes platforms, we're collecting images if they have, or lab data together with the samples from the patient and the family members on the RNA, the PAX gene tube, and with the genomic RNA too, for parents and when they're available, and from the patient. So probably
help me understand, will this be available for people in the relatively rural areas or in the relatively, how are you going to expand the outreach of genetics of rare diseases to those areas?
So right now they are in several areas of the country, not the rural. So they have these centers of reference for rare disorders in Brazil. They created about 17 of them.
Okay.
I think about 8 or 9 of them already have our approved protocol approved in the local IRB. In Brazil, you have to replicate the approvals in many different IRBs. But they do span from north to south, except the very up north, but from northeast. middle of the country, south region, southeast region, we have centers representing the patients. So they're gonna come from all different regions. We just have to stimulate those folks, or to engage and then to recruit.
I saw that there is one of those centers of excellence in Acre. Is that how it's pronounced? In the Amazon? Yes. That seems to be the only one that I could see in the whole Amazonas region. Is that a sufficient size to capture all these or is the region relatively not many people living there?
There's enough people to have another center, but unfortunately they did not create one there. Someone has to start the process locally.
Okay.
Request to the state health agency to be promoted, let's say, as a reference disease center. So there's a bureaucratic process, but someone has to start the process there. It's not centralized to say you are going to be the one becoming a reference center. So I have to stimulate folks to actually go ahead on those underrepresented regions and go and create one, so we can have more. They have more access to these kinds of technologies. And then
all the samples will come into your center. Where is your center now? Is it in
São Paulo? One of the Novo 6 in Recife, my hometown, and the other one in São Paulo, where the Albert Einstein Hospital is. I run both operations there and here and there. So,
okay.
But for these projects, most are going to be sequenced in São Paulo.
In São Paulo, okay, wonderful. Yes. And it's independent of the university. This is a separate effort, right?
Yeah, so I'm only working at the hospital right now. My lab was acquired by the hospital, and then we're just running the project and running the genomics service there at the hospital. So Manish.
Yeah, so there's a question from Christiana.
Chris, yes.
Would you interpret your transcriptome differently if you didn't have step 4 results? Great question. We truly would we would have not have noticed. We wouldn't have looked at the IL-12 receptor. Certainly, the splicing was at the 1th percentile, at the very, very top of a distribution of 400 healthy control individuals. Individuals who do not have immune phenotypes. So it would have maybe raised some attention, saying, "Oh, look at this. This is a really kind of aberrant in the distribution of the long and short isoforms." But beyond that, I'm not sure we would have gone looking any further. This is part of what we are calling a trinity. In immune deficiencies, you have to have an immunological phenotype, infections, autoimmunity, cancer, whatever. You have to have an immunological phenotype like T cells, B cells, signaling pathways, stimulation responses, whatever it may be. And you have to have a genomics phenotype, which is to say genomes, transcriptomes, splicing, whatever. And each of them contributes hypotheses towards what is the underlying cause. And you can sometimes be inspired purely from the clinical phenotype. Like this is a child who has eczema and a high IgE and maybe small platelets. Okay, well, we all know the answer to that one. But or a patient who has no B cells and was a boy and the uncle. Okay, you see, you can be inspired by some of the 2 of the 3 of the trinity. But in general, for the cases that we see on a day-to-day, week-to-week basis, like I saw a 23-month-old just now who has severely high fevers that come and go, exome negative. Like we're going to dive hard into this case. We have to look at interferon pathways. We have to look at inflammasome pathways. We have to do so much work to build a story around what we find genetically now, because all the interesting genes have already been, we don't have any rare variants in them. Now what? And so I think, Christian, yes, to answer your question, everything has to be considered to really to chase down the leads. Otherwise, you go crazy. There's too many leads to follow if you just use the exome or the genome or rare variants as your guide.
It's beautiful. And as you just cited and you showed, sometimes you can, even if you don't find, you can try to manipulate, right? You can do anti-IL-1, for example, in some of these kids with suspected autoinflammatory disorder and say,
exactly.
Oh, this is IL-1 driven, even if I don't know what it is. And so we have the chance now with this very specific drugs and tools, let's say, to a little bit, sometimes just experiment a little.
Absolutely true. Definitely. And in fact, that is the inspiration to figure out sometimes what is the genomic cause so that we can come up with a treatment. We face this problem in a non-public health system like ours, where to pay for that anti-IL-1 requires somebody to write a check, and no one wants to write that check unless they're convinced there's a variant. And so sometimes we will take I follow a man who has an NLRP3 variant, and we can just use that variant alone. Even if it's not proven to be pathogenic, that variant alone is enough to convince them to pay for the IL-1 blockade. problem solved. We can get him feeling better while we do the investigations. So genomics is just super important for just providing that extra firepower when you're on the phone with these thankless, faceless administrators who don't want to fund anything. We have to convince them to pay for our medicines, and we use the genomics sometimes as an excuse to convince them to move forward.
Thank you, Manish. I really appreciate your lecture and great insights here, and we hope we can collaborate in the future. So thanks a lot.
I appreciate it. Thank you. Have a good day, everyone.
Bye-bye.
Good evening. Bye.
Tchau, pessoal. E mês que vem a gente não tem aula. Em janeiro a gente vai ter do Dr. José Luiz Pedroso, especialista em neurogenética, referência no Brasil. E em março, Dr. Constantino Stratakis, from the NIH, falando de defeitos endocrinológicos, tá? Principalmente da adrenal. Bye-bye. Thank you. Have a good night.
Bye.
And a good day.